Hi, welcome back to another exciting year 13 video on photosynthesis. I hope you're all doing well. I'm doing all right, so don't worry about me. I'm doing all right in Casa de Polta. It's a lovely place, beautiful, beautiful. Anyway, so we'll start off with our first lesson into photosynthesis, and it's kind of a brief overview, kind of an introduction, bridging, if you like, between GCSE and Key Stage 5 Year 13 biology. So in the lesson, in the lessons PowerPoint I've sent you, I've suggested that you start off by trying to recall everything you can think of when it comes to your knowledge of GCSE photosynthesis. So hopefully you've done that, and now you're watching the video, and hopefully sort of things you might have remembered, of course, are the equation. Carbon dioxide plus water react to form glucose and oxygen with the use of light energy, and of course the balanced equation as well, and I probably will put that on the board in a bit as well. You've remembered the role of chlorophyll as it absorbs the light energy for that process. You hopefully remembered limiting factors, things that might affect the rate of photosynthesis, like temperature, CO2, light intensity, and so forth. And all those things are great. They're going to be really important to help act as a foundation, a kind of cornerstone for this much more complex Year 13 content. But it does get considerably more difficult, more complex, but I think fundamentally more rewarding and more interesting, because at GCSE level, it's sort of almost like this magic process, really. Carbon dioxide plus water makes glucose using sunlight energy. It doesn't really explain how that actually works on a kind of biochemical level, and that's something in the future lessons we'll really kind of delve into in depth. So the kind of starting point to look at here, I would say, is let's look at leaves. Leaves are extraordinary structures. I mean, my PhD was based on fossil leaves, so I became quite enamoured with them. I never really kind of appreciated them when I was your age, but I really did sort of appreciate how amazing they are. They're natural biological solar collectors, but not only that, they are able to absorb light, they're able to transport water and other assimilates from glucose, they can absorb CO2 from the air, so they are an exchange surface as well. So they're almost like a combination of plumbing, a solar panel, and lungs all in one structure, and they are quite extraordinary. So we've got to start thinking of some of the adaptations that we see with leaves. So your typical leaf, I'm going to draw a cheeky one here, like that, maybe something like a philodendron, something like that. You will notice that generally leaves are very broad, very wide, and that is to absorb as much incoming solar light energy as possible. They tend to be very thin, and that way, because the reason for that is because they absorb most of the light in the upper surface of the leaf, and we'll look at that when we look at leaf structure in a bit. And also to provide a much shorter diffusion distance for carbon dioxide to diffuse into those photosynthetic cells. So that's your general kind of overview of the leaf shape. You'll also notice veins they have. Now the veins are important because they contain xylem and phloem. Now the xylem are going to deliver water and mineral ions to those photosynthesizing cells like the Palisade cells to enable them as a raw kind of reactant for photosynthesis. The phloem are also going to transport away any assimilates, any products of photosynthesis like glucose. They can be used, say, in the roots or wherever there's non-sort of photosynthetic parts of the plant. So that's generally like our overall shape. Now leaves obviously vary according to their shape, and a lot of that actually correlates with the climate they're growing in. But let's delve a little bit deeper. So what I'm going to do is I'm going to put up an image of what the leaf structure looks like under a microscope. I'm going to talk your way through the key parts, the key tissue layers, and the kind of structural adaptations they have to help them photosynthesize. So the first thing to talk about is the uppermost layer of the leaf, which is the upper epidermis. So it's got a thin layer above it called the waxy cuticle that is a waterproofing layer. So that prevents any kind of evaporation of water. So that's going to reduce water loss from the plant. Only water should be able to escape from the stomata. The upper epidermis itself is very thin. It's a very transparent layer to allow the transmission of light to that photosynthetic layer below. So the Palisade cells, the most obvious things you'll see with these is they're very tall and thin, very tightly packed, and they're absolutely full of chloroplasts to maximise the amount of light absorbed for photosynthesis. And when you go to the layer below that, you will find that they've got the spongy mesophyll layer. These are very kind of large, rounded cells to increase the surface area. They've also got lots of air spaces to facilitate gas exchange, so to help absorb CO2 more rapidly from the air. Below that, we've got this lower epidermis, which is, again, kind of a waterproofing kind of outer layer to the leaf. And it's got lots of little holes in there, stomata, which allow gas exchange to occur between the inside of the leaf and its external environment, the air. And the stomata will obviously allow CO2 to diffuse in, water vapour, oxygen to diffuse out of the leaf. And obviously to control the size of the stomata, we have guard cells surrounding them, and also they're embedded within that lower epidermis layer, and they will open or close the stomata depending on the water balance situation of the plant. So if the plant is losing too much water, those guard cells will close the stomata to prevent water loss, and if the plant's got plenty of water, it will open them to allow carbon dioxide in. So that's kind of a nice little overview of kind of structural and functional adaptations of the leaf, and if I believe there should be a table that you can fill in your notes to help you get a nice sort of detail there. Don't hesitate to look at revision guides and textbooks as well as this video to help you out. The next thing to probably look at here is the chloroplasts again. So we have looked at them recently in our revision for year 12 content, but it's important that we have a little look back and understand their structure. So we'll have a look at the chloroplast next. Great, so here's my quick sketch of a chloroplast. Now hopefully you already know a lot of the features of it, but I'll run through them again just to kind of remind you. So we've got this outer envelope, so we've got this inner and outer membrane making up the envelope of our chloroplasts. Inside there we have this sort of fluid-filled cavity we call the stroma, and that's filled with lots of enzymes and proteins. And the stroma's primary role to aid photosynthesis, it carries out what we call the light independent reaction. Okay, I'll come back to that in a bit, don't worry. Then we've got these structures here. So this whole sort of stack of what looks a bit like coins or whatever stacked on top of each other is called a granum, okay, or grana plural. And each grana is made up of many thylakoid membranes. So each of those sort of coins, if you like, in that stack is a thylakoid. And that's where all the chlorophyll's located, and that is where the light-dependent reaction takes place for photosynthesis. Okay, these structures linking the different grana together is called intergranal lamellae, but you don't often get asked about that. One other feature, well, several other features you might see in here as well, of course, those smaller 70S ribosomes. There'll be some loose DNA. And you also might see quite commonly when you're looking at a cells type question, you might well see quite large circular structures which are starch drains, because obviously the chloroplasts are producing lots and lots of glucose. Now they don't want to keep it as glucose because obviously that will lower the water potential, causing water to move into the chloroplast by osmosis, and they will actually burst because they haven't got a wall around them. So instead it will store any excess glucose as starch drains. So just bear that in mind if you ever have to label something these kind of large. They will appear probably whitish because they're quite low density in electron micrograph. So that's a nice little overview of the chloroplast, and I mentioned a couple of new terms there. I've mentioned the light-dependent reaction and the light-independent reaction. Now we're going to spend an entire lesson on each of those because they are very, very complex. But that kind of starts to bring us forward and start to move away from year 11 and year 12 knowledge of photosynthesis towards that year 13. So photosynthesis is split into three kind of key parts. So I'll just get rid of the chloroplast. The first part is all about capturing light. Okay? And that is the job of the chlorophyll in those thylakoid membranes. Okay, so it's all about absorbing that light energy. And then what we're going to do with that light energy, because what photosynthesis is all about is converting light energy into chemical potential energy so that it can be actually used within cells. And not only just the plant cells, but even in my cells right now, the energy that I'm using in my cells has actually come from that process, because I'm a heterotroph like all animals. I have to eat other things to live, and we've got all of that potential energy originating from the light energy that plants have captured themselves. So that, in a way, we're, all life is somewhat solar powered in that respect, because we're getting our energy directly or indirectly from plants, and they're getting it directly from the sun, which I think is a really, really cool thing. So technically the energy has originally come from a fusion reaction in our local star, which I always find kind of mind-blowing. So the first stage there, chlorophyll capturing light. Then we carry out what we call the light-dependent reaction, so named because it depends on light energy. And that's a really cool little process, which is our next lesson, and it involves some quite complex biochemistry, and I find it kind of mind-blowing actually a little bit. I'm looking forward to teaching that one here. And that's all happening in the thylakoid membranes. And then lastly, we'll have the light-independent reaction, which doesn't require any light energy directly, but it does rely on two products that we make from the light dependent one, and that is where really we are actually producing the glucose. We're doing the carbon fixation going on, and that happens within the stroma, and that involves a complex metabolic pathway, sometimes called the Calvin cycle. So that's a series of enzyme controlled reactions with intermediate products between. So we'll get to that in our third lesson, hopefully next week. So hopefully that gives you kind of a nice little introduction to photosynthesis. Nothing too heavy yet. It's gonna get a little bit harder, and I suggest that you really take your time with these two to really get your head around. And hopefully the videos that I'm going to produce are going to help with that. Okay. So this is Dr. P signing off. Stay safe. Have a good time. Sun's starting to come out again. Have a cheeky one. Cheers.